And we continue with blood. So we've covered now the erythrocytes, all the categories of the leukocytes, and we move now on to platelets. So platelets are going to be the product of the same hemocytoblast cell that we've covered. So hemocytoblast, which gives rise to all the formed elements in a different pathway, then starts to become this cell we call the megakaryocyte. And the megakaryocyte basically is dropping crumbs, little cytoplasmic fragments, as they call them. So these little cytoplasmic fragments with little granules, but not a granulocyte. That's only for the leukocytes. Here we have these little bits of cytoplasm and enzymes and little clotting factors that we consider to be the platelets. So platelets, also known as the thrombocytes. As we mentioned, they're not actual cells, they're not complete cells. They're just little fragments, cytoplasmic fragments of these large megakaryocyte cells. Their primary role is not in transporting oxygen, not in immunity, but in hemostasis. Fancy way of saying blood clotting. So that's the role of these platelets, to help repair damaged vessels, to coagulate blood when it's necessary. So platelets have granules containing chemicals that act in the clotting process. It's a lot of chemicals there. So hemostasis, it's pretty straightforward. It's basically the clotting of the blood. But like with everything in the anatomy and physiology sort of discussion, everything gets complicated in vocabulary and more details here, right? So we're going to talk about some substances we're going to call clotting factors that are going to be released by the platelets. Clotting factors are also in the plasma, but they're initiated, their chemical reaction is initiated by some of these platelet substances. And we'll talk about those in a bit. A couple of things, I'm not going to get real detailed. I'll let you kind of navigate through that. But the spasm, vascular spasm, platelet plug formation, and coagulation. These are the three things that happen when we have an injury. So one, vascular spasm. So there is a constriction. Let's say we have damage to the blood vessel. That vessel constricts. Again, that smooth muscle that you remember from 2401. There's like a reflexive type of, something hits you, you kind of tense up. So that vessel constricts and it's triggered by different types of injuries. And we're going to talk about some of these, but it's that reflex initiated by the pain receptors, and that's what we term as the vascular spasm right there. So vascular spasm occurs, then the platelets start to make that little plug. And they make it seem like a very quick and easy step. It's a lot of steps, a lot of chemical reactions, it's a lot of metabolic pathways that go into causing that platelet plug right there. And then finally that coagulation to stop the loss of these formed elements there. So blood clotting, coagulation. In one word, what is the blood clot? The blood clot is fibrin. So that is the molecule in red, bold, exam type material. Fibrin is the actual clotting molecule, but fibrin doesn't exist in that state. There's going to be a lot of reactions that have to happen in order for fibrin then to be generated in the body. And a lot of these steps will involve these clotting factors, so coagulation factors, and that very fancy molecule that you know from, or I should say that element that you know, that ion that you know from 2401, calcium. Not much happens in the body without calcium being involved. All muscle contractions, and now we know clotting as well. I'll upload a little video that kind of goes through that hemostatic process. I'll steal it from YouTube, but let me continue onwards here. Clotting pathways. The book gets real detailed and real involved with the idea of clotting pathways. That emphasizes the extrinsic and the intrinsic pathway. Well, I'm gonna discuss them, but just know that they lead to the same thing, right? They start in different ways, but they kind of convert and they converge on one common molecule here, right? If we know the term extrinsic, it can exit out, so an intrinsic in. So what these pathways are is basically how or where the clotting is initiated. Is it initiated because of something outside of the blood, or is it initiated by something inside of the blood? And hopefully that makes sense in a bit. But once they are initiated, it triggers a domino effect. This converts this, converts this, converts that, and we end up going to that sort of common pathway. So class, coagulation factors, class. It's a bunch of them, right? So these are the factors: fibrinogen, thrombin, tissue factor, calcium, procelerin, and these are the numbers. And what makes it confusing is that the numbers are not in sequence in which they work. The numbers are basically there in the order that these things were discovered. So they were discovered in this sequence, but they probably work in a different sequence over here in the actual coagulation process. So most of them are going to be proteins, protein. Again, we have that calcium ion. Most of the clotting factors are also going to be derived from the liver. So this is significant, right? If somebody has any type of liver trauma, hepatitis or cirrhosis, you know, well, yeah, they're going to have other complications, but it's going to have an impact on the abundance of these clotting factors. So that's going to give them another complication there, right? You can read this over. It gives you a little bit of insight as to what these things do. But this is the big picture, right? And again, I'm not going to get specific. If you work in this field, phlebotomy, you got to know some more details here. But what I care that you know at this point is that whether we start here or here, we end up here with the prothrombin activator, right? So they're just two different paths to get to the same molecule. So when we talk about intrinsic versus extrinsic, so extrinsic basically is when we, let me see if we can make sense of this, right? So if we're inside of the blood vessel, right? So there's this blood vessel, this tube, we're at McDonald's Playland and we're in that tunnel, right? So we're kind of held inside of the tunnel and we're not being exposed to things on the outside of the tunnel unless that wall breaks and then we're exposed, right? So it's like, let's say you're inside your room and there's bricks outside of your wall, right? So those bricks and the wood and everything are protecting you from the stickers and the rocks and the mud and the dirt, whatever that's outside, right? So when are you going to be exposed to the stickers and the mud and the rocks? Well, you'd have to break through the brick wall. Yeah? Well, tissue factor we find, surprise, surprise, in the tissue of the body. And normally the blood is not exposed to tissue. Tissue is outside of the vessel. Blood is inside of the vessel. So there's damage. Now blood is being exposed to the outside factors, the tissue factors, and that triggers then the factor which sort of interacts with calcium, and we trigger this reaction. Intrinsic, well, if we're looking at the actual wall of the blood vessel, normally blood is, you know, rubbing up against that. But if that intrinsic, I should say, if that that lumen, that little layer of blood vessel is damaged, and we get into contact with the inside of the blood vessel, right? That's then the collagen fiber that triggers then the intrinsic pathway. So I don't know if that made sense or not, but the moral of the story, somehow, some way, intrinsically or extrinsically, tissue factor or collagen generated this, and we end up at prothrombin activator. So again, the extrinsic pathway on the right here, there's that important sort of tissue factor, or we call that factor 3. So the blood vessel has been ruptured. So now blood is in contact with tissue outside of that blood vessel. So the chemicals are extrinsic, outside of the blood. Intrinsic pathway is initiated by chemicals inside of the blood responding to collagen fibers. So like let's say that you break the wall in your room, you don't break the bricks to go outside, you just break the sheetrock or the panel, whatever. Behind that, there's probably insulation, right? That fiberglass insulation that's in there, or some type of insulation. So you mess with that, oh, and you get all itchy. So you're not technically outside, you didn't break through the brick wall, but you are now interacting with something that you don't normally interact with, right? And it causes a reaction. And that's the collagen sort of irritating, initiating the chemicals inside of the blood and causing our intrinsic pathway. So again, intrinsic, extrinsic should converge to the prothrombin activator. And this is important because we go onwards now to thrombin, and then thrombin will catalyze finally our fibrin mesh. And that's the goal. That's the, the big picture of this process. So fibrin. Fibrin is this sort of spiderweb looking protein. This molecule, very sticky. And its job is to actually function like a spiderweb. It's this mesh, this network. And as these little red blood cells try to, move across that, they get stuck. They generate like a traffic jam, and that basically stops the loss of these red blood cells. Plasma might still leak out for a bit, but that's okay. That's easy to replenish. The red blood cells will take a little bit more effort to replenish there. So the fibrin, spiderweb, fibrin mesh, trapping, adhering, sticking to the platelets. So that's the goal, right? This cross-linked fibrin mesh, right? So again, I hope all that made sense. All this pathway leading to prothrombin activator, and eventually then leading to fibrin. And again, I'm not going to ask these specific factors. I will address factor 8 in a little while. There's factor 8's role right here. But we got to make this mesh. After the mesh is done, that's great, good. We have then this clot. There's going to be a series of other reactions that then have to go in and remove that mesh. Otherwise, we then have a hindrance. We have some sort of structure in there that's going to then impede the flow of blood. So we got a clot. We make the fibrin. Fibrin does its job. Then we come in and do other repairs and remove the clot itself, right? And that little video might show a little bit of that in a simplified way. So just like everything, we know how it's supposed to work, but what happens when it doesn't work the way it's supposed to? So we have what we call thromboembolic conditions. So a thrombus is a clot that developed. It should probably have developed, but then it persisted after everything was repaired. So then now we have a barrier, an obstacle, something that's going to slow down the flow of blood in that localized area. We could have an embolus, or we call an embolism, which is a thrombus above, the thrombus that broke. And now that little blood clot is now traveling throughout the blood vessel. So the embolus is a clot, a thrombus, that breaks away from the vessel wall and floats freely. And you say, well, it doesn't seem like a big deal. Well, in the large vessels, it's not a big deal. If it gets to the small capillaries, let's say small capillaries of the brain, then you can run into problems like cerebral infarctions, strokes. You can run into, different, clogging of different little capillaries, so that it's not something that you want, right? We don't want traveling blood clots for sure. We can have, again, too much clotting, and then we can have the opposite, where we have the penias. We've talked about penia deficiency. So thrombocytopenia, not enough platelets to do their job, not enough factors possibly. If the liver is damaged, we don't have enough of the clotting factors. And the hemophilias are a genetic condition in which there's an absence of factor 8. So not enough factor 8, specifically factor 8, and then that's going to have a really negative impact on the progression of that intrinsic pathway. So again, if you're hemophiliac, too bad, so sad, that's just bad luck. Mom and dad gave you bad genes. For the others, these should be able to be reversed. If it's a bad liver, let's get the liver back where it needs to be. If it's a deficiency, we need to find out why the deficiency is in place. And again, if we don't have enough platelets, we always look for those megakaryocytes. Do we have enough of those megakaryocytes? Do we have enough of the hemocytoblasts? So we try to track back the developmental pattern and figure out what's going on, why we have then the deficiency. Is it a factor problem or it's an actual platelet deficiency? Something that I work with a lot. This is sort of my world, looking at the venomous reptiles. And I found a little video that I think you might find interesting on how basically venom works. We know that if the snake bites a little mouse or a rabbit, that mouse dies, the rabbit dies. But how did it die? What is the actual mechanism of death? Well, there's different venoms, different species. Some snakes cause extreme basically non-clotting. They don't allow the blood to clot. They destroy platelets. Others enhance the platelets, right? And they really stimulate the production of fibrin, and you have clotting throughout the whole body where you're not supposed to have clotting. So I'll try to upload that little video. I think you might find it interesting. If not, oh well. And there's a video. And with that, I don't want to get into that. There'll be another discussion or transfusions, right? So I'll stop it here and we'll continue.